Hello Biology 202 students, welcome to your online lecture. Today we will learn about the intriguing world of the male reproductive system, where millions of microscopic marathoners, also known as sperm, are created daily. Along our journey we will uncover how the male reproductive system functions, how sperm and testosterone are produced, and understand how the male reproductive system plays a role in reproduction. In today's lecture, we'll talk about the functions of the male reproductive system, the internal and external anatomy, the testes and the climate control of the testes, sperm production, sperm morphology, and hormone production, along with ejaculation, the prostate, erections, and methods for male contraception. The male reproductive system has two primary functions: the production of spermatozoa, also called sperm, and hormone production. Spermatozoa production is an exocrine function. This takes place in the testes, where sperm are produced, matured, and stored, and eventually delivered during ejaculation. Sperm production begins with spermatogenesis in the seminiferous tubules, followed by maturation in the epididymis, where sperm gain motility. The matured sperm are then stored in the epididymis until they are transported through the ductus deferens, also known as the vas deferens, and delivered during ejaculation. Alongside this, the system is responsible for hormone production, which is an endocrine function. Primarily, this involves the synthesis of testosterone by Leydig cells in the testes. Testosterone is essential for developing male secondary sexual characteristics, maintaining libido, and supporting overall reproductive health. So, how does all of this work? Well, luteinizing hormone, or LH, and follicle-stimulating hormone, or FSH, play crucial roles in regulating the male reproductive system. Luteinizing hormone, produced by the anterior pituitary, stimulates the Leydig cells in the testes to produce testosterone, a hormone essential for the development of male secondary sexual characteristics, and the process of spermatogenesis. Follicle-stimulating hormone, also secreted by the anterior pituitary, works by stimulating the Sertoli cells within the seminiferous tubules of the testes. These cells are responsible for supporting and nourishing developing sperm during spermatogenesis. Additionally, follicle-stimulating hormone promotes the production of androgen-binding proteins by Sertoli cells, which binds and concentrates testosterone within the seminiferous tubules, creating an optimal environment for sperm maturation. The male reproductive system's external structures are crucial for both reproductive and urinary functions. The scrotum is a protective pouch of skin that houses the testes, which are responsible for sperm and testosterone production. Running down the middle of the scrotum and along the underside of the penis is the raphe, a visible line marking where the two halves of the scrotum fuse during development. The penis is the organ used for sexual intercourse and urination. In uncircumcised males, the prepuce, or foreskin, covers the glans penis, the sensitive head of the penis. The corona is the ridge at the base of the glans where it meets the shaft. Finally, the urethra is the tube that runs through the penis, serving as the passageway for both urine and semen to exit the body. The internal anatomy of the male reproductive system is a complex network designed to produce, mature, and transport sperm from the testes to the outside world. It all begins in the testes, where sperm are produced within coiled structures called seminiferous tubules. From there, sperm move into the epididymis, a long coiled tube with three distinct sections. The head, where sperm enters, right here. Then we have the body, where sperm are matured, and then the tail, where they are stored until ejaculation. During ejaculation, the sperm travel through the ductus deferens, or vas deferens. This is a muscular tube that propels them forward via peristaltic contractions. The journey continues as the ductus deferens turns into the ampulla of the ductus deferens and meets the seminal vesicles, which add seminal fluid rich in fructose and other substances to nourish the sperm. This mixture, now semen, passes through the ejaculatory duct and enters the prostate gland, where additional fluids are added to enhance sperm motility and viability. The semen then flows through the prostatic urethra and passes by the bulbourethral glands, or Cowper's glands, which secrete a pre-ejaculatory fluid to neutralize any acidity in the urethra and female reproductive tract. Finally, the semen enters the penile urethra, surrounded by the corpus spongiosum and corpus cavernosa, which are the erectile tissue responsible for the penis's rigidity during erection. The semen is then expelled from the body during ejaculation. This intricate system ensures that sperm are well prepared for their journey and potential fertilization of an egg. Supporting the penis at the base are the bulb and the crus. These are roots of the corpus cavernosa, which anchor it to the pelvic bones and surrounding muscles. These structures play a crucial role during ejaculation and erection, helping to compress the veins and maintain the erection, as well as aid in the propulsion of semen during ejaculation. At the core of its anatomy are 3 cylindrical bodies of erectile tissue, 2 corpus cavernosa and 1 corpus spongiosum. The corpus cavernosa are paired structures running along the top of the penis, filled with vascular spaces that engorge with blood during arousal, leading to an erection. These are surrounded by a tough fibrous layer called the tunica albuginea, which helps maintain the rigidity of the penis during an erection by trapping blood within the erectile tissue. Beneath the corpus cavernosa lies the corpus spongiosum, which surrounds the urethra, the tube through which both urine and semen passes. Unlike the corpus cavernosa, the corpus spongiosum remains more pliable during an erection, ensuring that the urethra stays open and allows for the passage of semen during ejaculation. The distal end of the corpus spongiosum expands to form the glans penis, the sensitive bulbous structure at the tip of the penis. Again, the glans is often covered by the prepuce or foreskin in uncircumcised males, which can retract to expose the glans. The urethral meatus, also known as the external opening of the urethra, is located at the tip of the glans, where both urine and semen are expelled from the body. Now remember, there are 3 sections of the male urethra: the prostatic urethra, membranous urethra, and spongy or penile urethra. So this is a coronal section of the penis, and you can see both the left and right corpus cavernosum. You can also see the median septum, which separates the left and right, and then within the left and right corpus cavernosum, there are the deep arteries. These are supplying that blood that can fill the surrounding tissue to create an erection. And in your histology drawings, you'll be asked to also fit in the urethra, which is just below here, and it looks like this. Next, let's talk about the testes. The testes are housed within the scrotum. They are supported and protected by specialized muscles and connective tissue that ensure optimal conditions for sperm production. So the dartus muscle is a layer of smooth muscle within the scrotal wall, regulating the surface area of the scrotum by contracting in cold conditions to reduce heat loss and relaxing when it's warm to increase cooling. Then we have the cremaster muscle, which surrounds each testicle, raising or lowering the testes in response to temperature change, drawing them closer to the body for warmth or allowing them to hang lower to cool. We have the tunica albuginea, which is a dense fibrous layer that encases the testes, providing structural integrity and dividing them into lobes for efficient sperm production. Surrounding these structures is the spermatic cord, containing blood vessels like the pampiniform plexus that helps cool the arterial blood before reaching the testes, maintaining the ideal temperature for spermatogenesis, which is 35 degrees Celsius. So the spermatic cord is made of the ductus deferens, blood vessels, nerves, and the cremaster muscle. These tissues work in harmony to protect the testes and ensure they function effectively. For this lecture, I wanted to throw in some public health knowledge just to help out. So let's talk about testicular cancer. This is a relatively rare but serious condition that primarily affects younger men, typically between the ages of 15 and 35. It often presents as a lump or swelling in one of the testicles, though other symptoms might include a feeling of heaviness in the scrotum or a dull ache in the groin. Early detection is crucial, and men are encouraged to perform regular self-examinations to catch any changes early. To do a self-exam, it's best to do it after a warm shower when the scrotal skin is relaxed. Gently roll each testicle between your thumb and fingers, feeling for any lumps, bumps, or changes in size or consistency. A normal testicle should be smooth, firm, and free of any irregularities. If you notice any changes, it's important to see a healthcare provider promptly. Early detection significantly increases the chance of successful treatment. The histology of the testes reveals the intricate organization essential for sperm production and hormone synthesis. The testes are composed of numerous tightly coiled seminiferous tubules, where spermatogenesis occurs. These tubules are lined with a specialized epithelium containing Sertoli cells, which provide structural support and nourishment to develop sperm, as well as forming the blood-testis barrier to protect germ cells from the immune system. Between the seminiferous tubules lies clusters of Leydig cells responsible for producing testosterone, the hormone crucial for male reproductive function. So in this image, you can see a blood vessel there. Seminiferous tubule includes this whole tube. It kind of looks like a weird donut. Then we have sustenticular cells, which are also called Sertoli cells. Then we have our tubule lumen, and our interstitial cells, also called Leydig cells, are what produce testosterone. So within the donut, we have sperm production, and anything outside of the donut, we have testosterone production. And remember, there are lots and lots of these donuts in the testes. So sperm production, or spermatogenesis, is a complex process that occurs within the seminiferous tubules of the testes, and it's essential for male fertility. This process begins with the spermatogonia, the diploid stem cells located at the outermost layer of the seminiferous tubules. These cells undergo mitotic division to produce primary spermatocytes, which then enter meiosis, a specialized form of cell division that reduces the chromosome number by half. During the first meiotic division, primary spermatocytes become secondary spermatocytes, which contain a haploid set of chromosomes. These secondary spermatocytes quickly undergo the second meiotic division to form spermatotids, which are still haploid but not yet fully mature. Spermatotids then undergo a transformation process called spermiogenesis, where they differentiate into mature spermatozoa. This involves the development of a tell, or flagellum, for motility, the condensation of nuclear material into a streamlined head, and the formation of a acrosome, a cap-like structure containing enzymes necessary for penetrating the egg during fertilization. Throughout spermatogenesis, Sertoli cells within the seminiferous tubules provide structural support, nourishment, and regulatory signals to the developing sperm cells. These cells also form the blood-testis barrier, protecting the germ cells from the immune system and creating a controlled environment for spermatogenesis. The entire process of spermatogenesis takes about 64 to 72 days and results in the continuous production of millions of sperm daily, encouraging a steady supply of mature sperm ready for potential fertilization. So here's the morphology of a sperm. A typical sperm cell has a streamlined structure with the three main parts. A head, right here, which contains the genetic material and is capped by the acrosome, filled with an enzyme for penetrating the egg. Then there's a midpiece here, packed with mitochondria that provide the energy necessary for movement, and then a long whip-like tail, or flagellum, that propels the sperm forward. Abnormalities in any of these parts, such as the misshaped head, defective midpiece, or malformed tail, can impair the sperm's motility and its ability to successfully fertilize an egg, potentially leading to reduced fertility. However, it is common for some sperm to have abnormal morphology, and many men with such abnormalities can still achieve natural conception. So here are a few abnormalities. You can see differences in the head, differences in the midpiece. Some have two heads, some have two tails. Some tails don't look too great. Some midpieces don't look too great. And this is pretty average. You could probably find one of each in every male's ejaculate, but when there's a ton of these abnormalities, we get issues. Ejaculation is a complex process that involves the coordination of several different structures in the male reproductive system. It begins with the transport of sperm from the epididymis, where they are stored and matured, into the ductus deferens. During sexual arousal, smooth muscle contracts, propelling the sperm through the ductus deferens towards the ampulla, where they are mixed with fluids from the seminal vesicles. This mixture forms semen, which then passes through the ejaculatory ducts and into the prostate gland, where additional fluids are added to enhance sperm motility and viability. The semen then travels into the prostatic urethra and passes the bulbourethral glands, which secrete a pre-ejaculatory fluid that neutralizes the acidity of the urethra. Finally, the rhythmic contractions of the pelvic floor muscles and the urethra propel semen out of the body through the penis during ejaculation. So sperm are just the cells, and semen is the cells and the fluids from the seminal vesicles, the prostate, and the bulbourethral gland. Next, let's talk about the prostate. This is a small walnut-sized gland located just below the bladder in men, playing a role in reproductive health. It produces a fluid that makes up a significant portion of semen, helping to nourish and protect sperm during ejaculation. As men age, the prostate can become a focal point for health issues, most notably prostate cancer. Prostate cancer is one of the most common cancers in men, especially those over the age of 50. Early stages of prostate cancer often have no symptoms, making regular screening crucial for early detection. Common screening methods include the prostate-specific antigen, or PSA. It's a blood test, and also a digital rectal exam. Symptoms, when they do appear, might include difficulty urinating, frequent urinating, or blood in the urine. And you can see in this image on the right is a enlarged prostate, and you can see this portion here, which is the prostatic urethra, is kind of pinching off. So that's why it can get hard to urinate, and the enlargement can actually squeeze up on the bladder, causing for more frequent urination. And then cancers usually become more vascularized, so it's very easy to get a bleed in your prostate, so you could get blood in your urine. However, these symptoms can also be caused by other conditions like benign prostatic hyperplasia. If detected early, prostate cancer is often treatable with options ranging from active surveillance to surgery and radiation. Men should discuss their risk factors such as age, family history, and lifestyle with their health care provider to determine the best approach to screening and prevention. So we mentioned erections earlier. An erection is a physiological response involving the action of blood vessels, nerves, and erectile tissue in the penis. It begins with sexual arousal, which triggers the release of nitric oxide in the corpus cavernosa. This chemical signal causes the smooth muscle within the walls of the arteries that supply the corpus cavernosa to relax, leading to the increased blood flow into these erectile tissues. As the corpus cavernosa fill with blood, they expand and compress the veins that normally drain blood from the penis, trapping the blood and maintaining the erection. The corpus spongiosum, which surrounds the urethra, also becomes engorged but remains more pliable to protect the urethra and ensure the passage of semen. And this process is sustained by the autonomic nervous system and can be influenced by physiological and psychological factors. All right, our last topic is male contraception. So what can men do to prevent pregnancies? There are various methods of male contraceptions, each targeting different aspects of sperm production, transport, or activity. Barrier methods such as condoms are the most commonly used and work by physically preventing sperm from entering the female reproductive tract. Blocking methods like Vasogel Plan A include injecting a gel into the vas deferens, which blocks sperm from being ejaculated. Similar to a vasectomy, but reversible. Hormonal contraception, which aims to reduce sperm production by administering hormones that lower the levels of follicle-stimulating hormone and luteinizing hormone, thereby decreasing testosterone and sperm counts. Some approaches focus on inhibiting motility, preventing sperm from effectively reaching and fertilizing an egg. Spermicides are chemicals that are applied vaginally to kill sperm to render them inactive before they can reach the egg. Lastly, a vasectomy is a permanent surgical method where the vas deferens are cut or sealed, effectively preventing sperm from being part of the ejaculate. These options provide a range of choices for male contraception, varying in method, duration, and reversibility. Here's a conceptual question. If someone has a vasectomy, will they still have an ejaculation? They will, because of those seminal vesicles and prostatic fluid and bulbourethral fluid. So all that still comes out. There's just no sperm, as long as you've waited the proper amount of time for all the sperm to leave the vas deferens. All right, that is our lecture. Thank you so much for your time, and I'll see you on the next one.